So we'll start like we always start by looking at the warm-up. So let's take a look. All right, so again, remember this is material that's all going to be on your next main exam, which is going to be exactly 1 week from today. Right, you have exam 2 starts at 8:10 a.m. on 1 week from today. All right, sympathetic motor activity. So this is that kind of fight or flight. So on the pupil, it's going to dilate. The wide eyes, let in lots of light, try to see what's going on. Increases sweat. We talked about, in fact, sweat is pretty much just just a measure of sympathetic activity. Pupils are going to be push and pull. Parasympathetic constricts, whereas sympathetic dilates. Sweat glands are basically just more or less depending on more or less sympathetic activity. Heart rate speeds up and it suppresses your digestion. Basically, that's why we say like the rest and digest is parasympathetic. That's going to stimulate digestive processes, put your resources into kind of homeostasis and stuff. Sympathetic is on the other side. Somatic neurons, these are the ones that have one neuron all the way to their target organ. And at the target organ, what are they going to release? The target muscle, I should say.
Acetylcholine.
Acetylcholine, absolutely. Whereas autonomic, there's going to be two. You go first to the ganglion and then to the target muscle or organ. And what kind of neurotransmitters are released at the targets there?
Is it nicotinic?
No, actually. So first, what kind of so first we need neurotransmitters. What kind of neurotransmitters are going to be released by the autonomic motor neurons at their target? This should feel pretty intuitive by now.
Epinephrine and norepinephrine.
So there's epinephrine, norepinephrine is a possibility. If you're in the sympathetic side, what's another possibility?
Acetylcholine.
Yeah, acetylcholine, right? That's the main one for the parasympathetic. You definitely need to know this. This is at the core of, I talked about so much of the kind of pharmacopoeia, the drugs that are prescribed to, influence sympathetic or parasympathetic activity are going to be anticholinergic or anticholinergic agonists or antagonists or adrenergic agonists or antagonists. So you really need to kind of make sure you're straight with all this. How are the cholinergic receptors different in the somatic muscles versus the autonomic stimulated smooth muscles?
In the parasympathetic, it can either inhibit or it can be inhibitory or excitatory.
Yeah,
Whereas in the somatic, it's always excitatory.
that is correct. And what, at a deeper level even, what is causing that distinction? What's responsible for that behavior? What's different about the receptors, the cholinergic receptors on smooth muscles versus skeletal muscle? Maybe reach for it.
Is it
They're directly gated.
Say it again.
They're directly gated.
They're directly gated. Do you remember the name, like what we call them, what they're? Oh, so you guys, so this goes back. We introduced the idea of nicotinic versus muscarinic cholinergic receptors a while ago, and then we've been bringing them in different contexts. So definitely, especially given that your exam is 1 week from today, I highly recommend, finding the time to put into actually studying this stuff in a deeper level, because otherwise it's going to be, yeah, you're probably not going to do as well as you're hoping. This class takes a lot of time. This is not a class I've mentioned before. I'll mention again. This is not a class where you just kind of cram the weekend before the exam and you pull a rabbit out of a hat. Usually people end up just eating it when they try doing that. And that's not pretty for anybody. So make sure, yeah, I mean, I've noticed just at this point grading, I finished grading all the labs that have come in, and there's maybe like 2/3 of the class that are keeping up, and about a third of the class that I'm actually really concerned about passing the class, even who haven't turned in a majority of things. And that's not gonna, you can't, this class is a class you do need to keep up with. You do need to pass both the lecture and the lab sides too. You can't just like, okay, I'll try to pass the lecture exam and that will balance out my F on not doing any labs. So please, it feels bad for me as well as for you to see people falling through the cracks. And if you need help, reach out. But at some point, I can't do anything if nothing's coming back and I don't know what's going on. So let's continue on here. Blocking adrenergic receptors on the heart. So this one people, this gets a little more into a subtlety, but it's important that you understand the subtlety here. So blocking adrenergic receptors, it will not directly slow down the heart, right? What it's doing is it's blocking the sympathetic neurons that would speed up the heart. So if you have an adrenergic antagonist, something that blocks adrenergic receptors, it prevents the heart from speeding up in response to something. But it's not going to directly slow down the heart rate, because if you want to directly slow down the heart rate, you need to do parasympathetic activation. That's what's going to slow down the heart rate. Taking a beta blocker means that if you get nervous, your heart's not going to speed up because those sympathetic messages are never going to get to the heart to speed it up. But it's not necessarily slowing it down. It's the kind of tonic parasympathetic innervation from the vagus nerve that's keeping the heart slower. So yeah, make sure that distinction is clear. It's similar like the eye drops they give you when you're getting an eye exam. What kind of a drug would you want? They want to shine a light into your eye, but you don't want the pupil to constrict. You want the pupil to stay open so you can like shine a light in and look around. You know, what kind of a drug would you give to prevent the pupil from constricting?
You would give eye drops that have nicotinic antagonistic.
Yeah, exactly. So a nicotinic, or I should say cholinergic antagonist. Yeah, because actually they're not going to be nicotinic; they're going to be muscarinic. But it would be cholinergic antagonist, something that blocks acetylcholine, and that's going to make, when you shine a light in the eye and the parasympathetic innervation turns on to constrict the pupil, it's going to stop that message so the pupil stays dilated, right? You could say like, oh, give them a adrenergic agonist, but then that means you're fighting, right? Your parasympathetic is trying to pull it down, and then you're giving them something that's pulling and trying to pull it open. That's not very graceful to do. You just stop the response that's gonna constrict the pupil, and that's gonna accomplish what you want without having the system fighting itself. Okay. The neuromuscular junction, where somatic motor neurons innervate skeletal muscles, are characterized by the neurotransmitter. What? So which neurotransmitter? You know, it's acetylcholine. Everybody got that? And the receptor is going to be the nicotinic cholinergic. So here people got it. So people were thinking about it, but I guess in the other context, people were getting kind of having trouble putting it all together. So finally, all senses. Yeah, a lot of people, yeah, so most people got this. All senses except for olfaction or smell will synapse in the thalamus before continuing. Smell is the one that's different, that has paths direct into other subcortical regions. So any questions about all this stuff? I can stop sharing here. Right, all of this is this, and actually all of the other sense we're talking about a lot, introducing general senses, olfaction and gustation last time as well. So are there any questions about any of that material? Because we have a lot to cover this week. We're going to finish up today. We're going to finish up the nervous system. We'll do more on the eyes and the ears, and then we'll move on into the muscle system. And we're going to get deeper into how muscles work, how muscles are kind of regulated, both skeletal muscle, smooth muscle, and actually cardiac muscle will be kind of like the segue taking us into the cardiovascular system.
Good. I had one question. So everything from now on will not be on this next exam. Is that correct?
No, that's not true. Everything through this week will be on this exam. The exam will go up through the muscles. Yeah, so no, definitely, everybody should realize this exam is going to cover all of the nervous system, including the stuff we're going to finish today, plus the muscles that we're going to be covering, hopefully at the end of today into Thursday. In my dreams, we'll actually finish the muscles and start the cardiovascular system by the end of this week. And if that happens, I'll tell you where the line is and where we're starting the new material that won't be on the exam. Cardiovascular will be on exam 3, but nervous system and muscles are on exam 2. Right? That's why I was telling you, make sure you keep, even though it's break, I worked almost every day over break. You got to kind of keep up on this stuff because there's just more stuff. So making sure you're comfortable with the stuff that's come before, and then you add the new stuff on. So then you're not like scrambling. All right. So it's time to finish the nervous system, starring the special senses. All right. Whoa. Hold on, we need to mute somebody. Okay. I should say Gesundheit or something. All right, special senses continued. All right, so we have covered olfaction. It's supposed to be a nose. Again, what is the stimulus for olfaction?
Isn't it like chemicals?
It's chemicals, right? It's basically a way of detecting what different kind of chemicals are floating around in the air. You had those special olfactory cells in the kind of roof of the nasal cavity. And those little olfactory cells had little receptors that could bind different kinds of molecules. And depending on what molecule, it would, create, basically open some channels, create depolarizations, send messages up to the brain saying, again, the smell of roses or the smell of onions or the smell of mint or the smell of poop are just your subjective experience of having different kinds of molecules floating up into your nasal cavity and binding these receptors up in here. Right, which is always a little, I remember the first time I kind of learned about that, it was a little disconcerting realizing that, like, if it's like, oh my God, I stepped in dog poop. If you're smelling dog poop, that means there's little dog poop molecules floating around up inside your nasal cavity, binding your olfactory cells. So, but don't think about it. What about taste? Gustation. What is the stimulus for gustation, for the taste buds? Anybody?
Chemicals as well.
Yeah, chemicals as well, except this time they're dissolved in your saliva and is a more limited group of categories. Like smell is got this huge array of different kinds of molecules that it can bind and recognize and gives you a huge range of different kinds of smells that you experience. Whereas taste has the 4 or 5 basic tastes, like sweet being simple carbohydrates, sour being low pH, bitter being alkaloids, salt being salt, umami being like monosodium glutamate and stuff. So they're both chemoreceptors, and we're gonna now, again, we covered those, but I just wanted to kind of make sure you remember what they are. And again, at the core, how does sensory transduction work for these guys? How would you accomplish sensory transduction either for smell or for taste? Like how could a taste bud know that there is sugar dissolved in your saliva? And how could it convert that into some electrical signal, which is what transduction is?
Aren't there receptors for it?
Yeah, exactly. It's just going to be like receptor.
Exteroreceptors.
Yeah, there's going to be, or I mean, we would use the word exteroreceptor for the more broad term, like the taste bud is an exteroreceptor, but there's also just G protein coupled receptors in the membrane of those little taste cells that bind the sugar molecules, open channels, and create depolarizations that ultimately result in action potentials getting sent to the brain. So, yeah, I mean, it's a little confusing. The word receptor, sometimes we use receptor meaning like the whole organ, like the eyeball is a receptor or the taste bud is a receptor. But in this case, I'm meaning receptor like the little membrane receptors, those membrane proteins that bind ligands and usually open channels or do different things. So yeah, the same word obviously means different things in different contexts. Okie dokie. So now we need to do the other two special senses, vision and hearing, which are going to be more complicated. We've already covered a lot of vision in terms of like how you focus and things like that. So let's do vision a little more. We're gonna go deeper though into the retina where the actual sensory transduction is happening. We're also gonna go a little deeper into some of the physiology that kind of keeps the structures supplied with nutrients and stuff. Because so if we look at an eyeball, this is like the eyeball from the front. And then we're gonna do kind of a cross cut through it. And we'll get that picture that we've been drawing in lab a bunch. So this is like the cross section of the eye. And again, we have the iris. Iris, which comes from the root meaning rainbow, because it comes in all these different colors. And again, the iris is mainly smooth muscle fibers. Some of the smooth muscle fibers go in a more longitudinal direction. Right, so if those are activated, those are going to be activated by sympathetic. That's going to dilate your pupil, going to open up the iris, let more light in. Some of the muscle fibers go in the concentric direction, just concentric circles. Those are going to be triggered by the parasympathetic. If those are contracting, it's going to squeeze down and make the pupil smaller. Again, the pupil, just a little hole that allows the light to enter into the eyeball. It only looks black because it's dark in there, right? That's why you get red eye when you get a flash, because then you're actually illuminating all the blood vessels and everything that are actually in the back of the eyeball. So the smooth muscle of the iris, that is part of what we call like the intrinsic eye muscles. Intrinsic meaning kind of inside. You know, there are also extrinsic eye muscles that move your eyeball around. It's like if you're looking around, you got to have muscles to rotate your eyeballs around. That's a different kind of muscle. Those are skeletal muscle. Those are all, you know, somatic. These guys are autonomic. And so you have the ones that operate the iris, and then you also have the ones that operate the lens. And that's operated by these things called the ciliary muscles. And the ciliary muscles can attach to the lens with these little, they're called suspensory ligaments. the system is a little counterintuitive. The reality is the muscles actually kind of pull off to the side and release tension on the eye. So when the eye is relaxed, the lens is actually going to be at its most thin. And as you contract these muscles, it kind of releases the tension on the lens, and the lens kind of pooches out more and becomes more and more round. So you have to use energy, use the muscles. When the muscles contract, it releases tension on the lens and allows it to become more curved. Right. So if your eyes are emmetropic, if you just relax your eyes and don't work at all, then your eyes will be perfect to look far away and have everything in focus. And then that accommodation we talked about is contracting these ciliary muscles, which then pulls back, kind of releases that tension, allows the lens to bulge out more. So you have to work. You've got to work to actually make the lens bulge and focus on closer things. Both the lens and then the cornea is like the clear window that goes into the eye. Both of them have to be completely clear, and yet they have cells, epithelial cells that need oxygen and nutrients. Like the lens is covered with this special lens epithelium that makes these special crystalline proteins that fold in this highly regular way to make up the bulk of the lens. So the lens and the cornea, they need nutrients. You don't want blood vessels on them because then you're going to be looking out through blood vessels. So there's going to be another system, kind of like similar to what we saw with the CSF, where there's going to be this aqueous humor, which is being produced and flows around and provides the nutrients to these clear structures. So let's talk about that. So the ciliary muscles are here. They are part of a larger structure, which we just call the ciliary bodies. And these produce what's called aqueous humor, which is a lot like CSF. Let me spell correctly. So you've got this aqueous humor that's being produced that kind of irrigates all around in here. And providing nutrients to the cells of the lens and the cornea. The back of the eyeball is filled with stuff that's more jelly-like that helps it hold its form. So the back here, this is, maybe I'll make it in green. This back part, instead of being filled with the more just liquidy fluid, is filled with something that's more gelatinous called vitreous humor. Vitreous means glass, so it's kind of jelly-like. For those of you who've dissected cow or sheep eyes in anatomy, you've seen it. If you open up an eyeball, it looks like a big gooey gumdrop that falls out of the eye. But it's permeated. This aqueous humor here irrigates all around this anterior part of the eye, and also kind of irrigates and permeates through all in there as well, because it's just this kind of wet jelly. So similar to the brain, where you're constantly making CSF, here in the ciliary bodies, we're constantly producing aqueous humor. We obviously have to drain it, or else the eye is going to start building up too much pressure inside. So there's a drainage system, and the drainage system is right at the boundary between This is the iris here and the cornea. So I'm kind of drawing here. There's like a little canal. It would be like a big toroid, like a big donut that goes all the way around. It's called the canal, one word of it, canal of Schlemm. That is draining this aqueous humor. So it's being produced, it's being drained constantly. And it ultimately just drains into the same system that the CSF drains into and ends up going back down your jugular veins. So again, it's another thing, this very plasma-like stuff that then just dumps back into the bloodstream. What is the pathological condition if that drainage is blocked and you actually start building up pressure in the eye?
Glaucoma.
Yeah, that's glaucoma. And glaucoma can be really dangerous because it actually starts impacting the ability of the retina, the sensory cells, to stay healthy and get their nutrition and all that. So you can ultimately go blind from that. When people go in to get eye exams, they don't just check your focus. They also check your pressure in your eyes, because that's another important indicator of your eyes functioning well and being healthy. What other things? You can have clouding of the lenses and everything just looks kind of milky. What do we call that?
Cataracts.
Cataracts. So cataracts are when you have the milky, kind of the clouding of the lenses. Again, glaucoma is when you have a buildup of pressure because you have inadequate drainage of the aqueous humor. It's interesting when you have cataracts, they will go and they'll take out the lens and they'll put in a new lens. What is going to be the problem with putting in just a artificial lens there?
I mean, your body wouldn't be used to it.
No, they pick materials that are not going to be rejected by your body, so that part isn't a problem.
And an
Not contract it and relax the muscles and change the lens shape.
Yeah, so you're not going to be able to change the lens shape. You're not going to be able to accommodate like you used to. So, when people get cataract surgeries, typically they have to choose. Do you want, actually, there's three choices. Sometimes people get them optimized for far vision, then they have to use glasses to read close, or they have them optimized for near vision, use glasses to look far, or I know people actually who get one of each. And so they have one eye that is just permanent far vision, another eye that's permanent close vision, and their brain after a little while just automatically just focuses, takes in the image from the appropriate eye depending what they're trying to pay attention to. I mean, that sounds really unpleasant to me, but it definitely works for some people. So, but yeah, no, it can be, I don't know if you know people, hopefully none of you have had to deal with cataracts. If you know people, like it can be like amazing. Like my mom was just like terrified of going in for surgery, and then like later that afternoon, she was just like over the moon with how her vision had returned. It was crazy. All right, so this is kind of like bigger picture of the eyeball you should know about. The other stuff we're going to talk about is on the more sensory transduction side, looking more at the retina, where the actual light is being converted into electrical signals. And again, that is the retina, which is basically all along kind of the back of the eyeball here. And it might be easier just to actually show this on the PowerPoint because they have good pictures of it. So I'm going to go to that right now. And I guess before we get deeper into this, I do want to spend a little more time talking about the stimulus for vision, especially if people haven't like done much physics up to this point. You know, I think it's interesting to like think about what is light? What are we actually sensing? Like with, you know, taste buds, you know, sugar molecules, so you can kind of get a better sense of like, oh, it's a thing, a sugar molecule. But light, what is light? Light's a little more esoteric in some ways, in a lot of ways. Like what is visible light? What are we actually sensing?
Photons?
Yeah, photons. And even what a photon is it's kind of bizarre. You know, it's these, you can think of photons as like little packets of light energy, but then what is light energy? Light energy are these undulating electromagnetic fields. And what we think of as light energy is just a small little band of frequencies of electromagnetic waves, electromagnetic radiation that just kind of flows through everything. And like if you think about sound waves, like sound frequencies, right? There's like low, you know, those are all like sound, but just different frequencies. So when you think of the electromagnetic waves, as you go from like lower frequency to higher frequency, you know, rather than just being, you know, thinking about, oh, it's a different sound, but just different frequency, we give them very different names even. So like right here, this is showing like the electromagnetic spectrum. You know, the visible light, this is the wavelength. This is in nanometers, like billionths of a meter. You know, the ones that have longer wavelengths means they're lower frequency, we perceive as red. As the frequency gets higher and higher, instead of hearing a higher pitch sound, we see the colors towards purple in the rainbow. But this little band of frequencies of electromagnetic radiation that our eyes respond to and we perceive as light, it's just a little piece of this much larger thing. If you go faster frequency than purple, you get into the ultraviolet. If you go even faster than that, you're in X-rays. Faster than that, you're in gamma rays. If you slow down below red, infrared, we just perceive that as heat instead of as light now. And then microwaves. You know, microwaves are the same stuff as the rainbow. They're just slower frequency, and they happen to entrain water molecules and make them vibrate, which is good for heating up your food. Radio waves, it's the same stuff as visible light. It's just much slower, much larger wavelengths. So just to kind of put that in context, what light is this undulating electrical and magnetic fields permeating through space. You know, the majority of the energy, electromagnetic energy produced by the sun that's landing on the earth is in the range that's, you know, what we think of, that we perceive as the rainbow, which is I think why our eyes evolved to like optimize to pick up those frequencies. But just kind of, I think it's, yeah, it's useful to remember what light actually is. It's this energy. And again, it's being picked up by the retina. So let me like, I'm gonna go slideshow, current slide, pointer options. All right, so here's the retina. kind of going along the back here of the eyeball. And again, the optic nerve, this is going to the brain. And we talked about right where the optic nerve leaves, that's going to be where you have the blind spot, the optic disc, where there are actually no rods and cones, no sensory transduction. This here, this is the more detail of what the retina actually looks like. And there are going to be three main layers. There's this layer of rods and cones that are the actual phototransductors, the things that convert light to electrical signals that we're going to look at in much more detail in a few moments. And then there's a lot of other cells that, you know, I'll give them names. In the interest of time for our class, we can't go into much more detail. There's the next layer called the bipolar cells. The next layer are called the ganglion cells. The ganglion cells are the ones that actually create the optic nerves. Like these axons from the ganglion cells are what actually become the optic nerve and go to the brain. There are also things called horizontal cells in there that are actually important, and amacrine cells. And again, in the interest of time, I think I just wrote horizontal. Excuse me, horizontal cells, amacrine cells. So there are miles and miles and miles of axons and stuff intertwining in the retina. The retina is very different from the other kind of transducting elements we've seen, receptors. Developmentally, in fact, remember the diencephalon part from the forebrain. In addition to becoming the thalamus, hypothalamus, and epithalamus, I didn't really bring it in, but you also have the optic cups, which come off the diencephalon, which then actually migrate out and become the retina of your eyeball. So the retina is actually kind of a little out-pocketing of the brain and does a lot of kind of more complex pre-processing of the visual imagery before the message even goes down to the brain. Your book actually has a nice little thing talking about like lateral inhibition to increase contrast and things like that. And again, there's a lot of fun stuff we can talk about, but I also want to make sure we get through the real core stuff. So I'm just going to kind of leave it there that these other cells in here are doing kind of interesting things, enhancing contrast, things like that. The retina is not just about sensory transduction. The rods and cones are the ones we're going to focus on. So let's do that right now. And I should also mention that it's very non-intuitive that the rods and the cones are on the back of the retina, right? Because the light, if I go get my little pointer back. You know, the light's coming in. If we look at this, the light has to go through the nerves, through the ganglion cells, through the bipolar cells, and finally get to the photoreceptors. So luckily these cells are clear, but it's, the light doesn't get to the rods and cones until it actually goes through all that other cells. When you get to the Fovea Centralis, which we talked about, the place where you have most acute vision, the cells and the axons are kind of folded out to the side a bit to minimize the thickness of the distorting layers of cells that the light has to pass through before it actually gets to the cones that are sitting there in the back. Okay. So this is the eyeball again. I'm going to talk about the retina again. Again, the retina is just kind of sitting all along the back. It's those 3 layers with all those other horizontal cells and amacrine cells. Again, for our purposes, we're going to focus in more on the rods and cones, the photoreceptors. So let's talk about rods versus cones. We've talked about them before, but we're going to talk about them in more detail now. So again, they're both photoreceptors. They both take light energy and convert it into an electrical signal that gets sent to the brain. So these are both photoreceptors. The core mechanics of how they work is the same. We'll see in a few moments. How are they different from each other? We've talked about this in lab a bit.
One picks up on light and like differentiates between light and dark, and one sees color.
Yeah, so rods are monochromatic. They can distinguish between light and dark if there's the presence of light, but they can't tell you what color. They are kind of maximized to respond to wavelengths that are around the green or so. That's why, in fact, why you use red as a safe light in the dark room or something, because the rods don't even absorb that at all, so they don't end up getting bleached by red light. Cones, on the other hand, detect color because we had 3 different kinds of cones. If you remember, if I looked at, response, and I looked at kind of wavelength, or I should say, let's say frequency. I like thinking of frequency because it's more intuitive for me. Frequency of the light, red. Remember Roy G. Biv? Everybody, you probably learned Roy G. Biv. Red, orange, yellow, green, blue, indigo, violet. That's just the rainbow. And there are cones that respond primarily to red, cones that respond primarily to the green, cones that respond more over to here. And depending on what combination of these that you're mixing up, you'll detect different colors. All of them together we mentioned was white because luminance mixes in a different way than pigments. So these would be like the cones. There's 3 different kinds of cones. If you're missing one because due to a genetic problem, you're going to be colorblind. You're going to have an inability to distinguish certain colors. The rods are somewhere around the green, so they detect if there's color or not, but they can't tell you what color, what flavor, what frequency it is because there's not different versions of them. So that's one big difference between rods and cones is just rods can only detect presence or absence of light, whereas cones can tell you also what color it is, what frequency it is. What are other differences between rods and cones?
Acuity. Cones have high acuity.
Yeah, so cones have high acuity, meaning you can resolve, like acuity means if there's 2 dots, I can tell it's 2 dots and not smeared out into 1 dot. Kind of like that 2-point discrimination that we saw on our skin, right? How close can you put those 2 dots and still know it's 2 dots instead of 1 dot? Like rods, there's something there, but I can't quite tell you what it is. Or I know that person's got their hand up, but I don't know how many fingers they're holding up right now. Or I know this thing's a book, but I couldn't tell you for a million years what the words say on the page, right? So you see something, but you don't have the detail of it. Part of that happens Well, actually, let's add a few more things and you'll see how this happens. Because rods in and of themselves aren't necessarily low acuity. They're just photodetectors. It's how they're wired up that is going to be a big part of how they respond. What's another difference between rods and cones?
Isn't there a higher concentration of cones right at the back at the Fovea Centralis?
Mm-hmm. So right in the middle here, what we call the Fovea Centralis, this is going to be mainly cones. Whereas the rods tend to be distributed around what we call the periphery. So the rods, periphery of retina. Cones are in the Fovea Centralis. So when you look straight at something, that image, that kind of sharp, clear image you see of it, that is landing on the cones of the Fovea Centralis. As soon as you get farther away from the center, like, again, right now you're staring at me on your screen probably. Like, put your hands on either side of your head and wiggle your fingers, right? You'll be able to see, like, there's hands wiggling, but you can't tell even how many fingers or if you're wearing nail polish or whatever, right? It's just, there's something there, but you don't got the acuity. You don't have the detail of what it is. The only thing that you can see with detail is what you're staring right at, what you're foveating on. Again, there's so much cool stuff we can talk about, how you try to, because you always kind of have this sense that you see the world around you in high definition, but you actually only see the stuff right in front of you that you're staring right at in high definition. Everything else is a blur. But we kind of keep recreating our sense of what everything looks like around us. And you can play around with that and you can do these really disconcerting illusions like they call change blindness and stuff, where you can have the world change in dramatic ways around you, and if you're not staring right at it as it happens, you have no idea anything's getting weird. So periphery of retina, high acuity. So another difference is cones respond to bright light. So cones will not function in low light. They need bright light to be functioning. Rods, on the other hand, can will work in low light. So when you're out walking out under the moonlight, you're in rod vision. That's why things look monochromatic, right? If you're walking around in the moonlight, you don't have a lot of vibrant colors on everything. If you've ever taken a very long exposure camera picture of things under the moonlight, or actually with these new cameras now that do like the low light thing where they just leave their sensors open and take in, it's very low light, but they just take it in long enough that they get enough to get a good picture. It's really trippy because the colors are still there. Trees are still green at night. It's just that when you're looking at a tree with rod vision, which are the only ones that are being functional in low light, they don't have the ability to tell you that the tree is green, right? The tree doesn't turn gray at night. The tree stays green at night. It's just your ability to detect green goes away. That's again why these kind of fancier like iPhones now can like take a picture of a tree at night and it looks green. It's like, whoa. So part of the way the rods and cones do their thing, the rods tend to have a lot of kind of convergence. Like a lot of rods will all end up becoming one axon, leaving the eye down the optic nerve. We talked about the ganglion cells doing the axon. So one ganglion cell is getting information from lots of rods. So even if a few photons are falling in here, there's a big array that can detect that something happened and say something happened, but I can't really tell you exactly where it happened. I don't know which one of these rods was the one that actually noticed something. With the cones, each cone basically gets its own highway, its own axon going off to the brain. So that means you have much more resolution. I can tell, is it this cone or this cone or this cone? So I have a lot of acuity, a lot of detail, but I need a lot stronger of a signal to go in to actually activate that cone strongly enough that it actually sends a message that I can notice. So cones are going to be high acuity, but they need brighter light to function. And again, also they can detect color. Rods, monochromatic, low acuity, but they can give you a visual, give you visual information, even if it's very dim. And I should, for whatever reason, people sometimes on these exam questions, like, what are differences between rods and cones? People, well, cones see in the light and rods see in the dark. You know, it's like, you know, you can't see in the dark, right? If there's no light at all, then there's nothing to see. So rods don't see in the dark. What they do is they see in very low levels of light. They see in the dim, right? You know, when we say it's like dark outside, we don't really mean it's pitch black. We mean that it's, you know, very low levels compared to like the sun. So normally if we were in classrooms, you know, I have these blackout shades and I actually run through the thing where you actually go back and forth between rod vision and cone vision so you can actually experience it. Because we can't do that, I actually have a little homework thing for you to do, which I'll just tell you right now. And the basic idea is you need to find some place in your house. It could be a closet, could be like the living room late at night with all of the shades drawn or something. But it has to be something that you can lower the light down really far. There has to be a little bit of light left or else you can't see anything, but it can't be, you know, if you're still seeing a lot of color, it's too bright. Because if you're seeing color, that means the cones are going. And I should also mention the rods, if you have bright light, they all get bleached. We're going to talk about this a little more in a few moments, but basically rods are going to need time to recover if it's been bright, right? So when you first turn off the lights, the rods are all what we call bleached out. They need time to recover and come back online. When it's really bright, they all go blah, and then they slowly kind of, well, okay, let's get ready to function here. So when you first turn off the lights, you might not see much of anything because the cones aren't functioning, but the rods haven't come online yet. We have what's called this dark adaptation. Like, you know, rods come back online after they've been in bright light and all got bleached. So this little homework thing, let me get it now. Okay, modules. So So this here, I'm posting it now, so it exists. First-hand experience of rods versus cones. So I have it. I put here due March 11th. Although I would do it before then. So basically, it's just kind of reviewing the stuff we're talking about. Two kinds of photoreceptors, rods and cones. La la la. For you to do. I really want you to, I don't think there's any way I can talk about it, you can memorize it, but when you really experience it, it's like, oh, that's like, I see it. It totally makes sense. So the first thing you do, cone vision. To do cone vision, all you need to do is be in the bright. So turn on the lights so it's nice and bright. You're using your cones. And again, the things I want you to pay attention to, I want you to write down, describe your experience of color. Do you see colors? And your visual acuity. Like I'd say it's worth just having a book in front of you because that's a good way to tell if your visual acuity is high or low. If you look at a book and you can read the letters, you know you've got good visual acuity, assuming you're wearing your glasses or whatever. Second, go into rod vision. Turn down the lights way down low. It sounds like a song or something. Note that there has to be a minimal amount of light or you won't see anything, but it should be very faint. In this condition, your cones will no longer be responding. At first, your rods will still be bleached from the light, so you won't see much of anything. But after about 5 minutes, you'll probably start seeing lots of stuff. So once you've waited 5 minutes, you're going to describe again what you're seeing. Your experience of color, your experience of visual acuity. Can you read a book that's right in front of your nose? And kind of hint, if you still see colors pretty well, then you're not really in cone vision. You're not in rod vision. So you got to kind of try to lower the light a little more. Then once you have kind of noticed both color and visual acuity under rods, flip the lights back on. This could be a little disconcerting. It's like, whoa, especially after you've been in the really dim light, and just describe again now what's happened to the color and to your visual acuity and your sharpness of vision. So, submitting a text entry box, but I want you to do this. This is worth points because I really think it's one of the ways to really kind of solidify this understanding of rods versus cones is to really see they're radically different in how you perceive the world. So this is another assignment. So that's, I should ask actually, are there any questions about kind of the expectations on that? No. Okay. Again, normally we could just do this in lab, but we can't do it in lab. So you'll have to find a closet. So what we're going to do now is talk about what exactly is happening in a rod and a cone that convert light energy into an electrical signal. And it's going to just be using concepts you've already been familiar with. The idea of G protein coupled receptors and phosphodiesterase and all that kind of stuff, right? Like I mentioned back at the beginning, all these concepts all layer on top of each other. So we are going to talk about phototransduction. So how does a rod and a cone actually work? And I'll be mentioning basically a rod here, but rods and cones do their thing the same basic way. They have a slight different shape. They use different versions of what we call opsins that absorb light energy at different frequencies. But the story I'm going to tell you right now is basically the same, whether or not you're looking at a rod or a cone, how you convert light into an electrical signal. So let's do it. And like second messenger pathways, which this is, it's going to be a little convoluted, right? It's going to be this cascade of events. This goes to this, goes to this, goes to this. So you're going to have to kind of keep track of this stuff. Probably like writing it out and making sure that you understand like what happens when is going to be important. This isn't just like remembering, squirrels live in trees and gophers live in holes. It's going to be lots of interlocking pieces. So this would be like a rod. It's got this part here, it's called the outer segment. This part called the inner segment. So I'm going to add in some of the main parts and pieces that we're going to be doing, and then I'll talk about how they all fit in together. So in this outer segment, we have these membrane proteins called opsins that are tied into a little thing called retinal. So this thing that I just drew here, this is opsin plus retinal, which we'll talk about in more detail. And there's also this thing called transducin, which is basically a G protein. An opsin is actually a G protein coupled receptor. But as we'll see, every receptor we've seen so far got triggered by a ligand binding to it. This thing is going to get triggered by light hitting this complex, hitting the retinal opsin complex. So that's up what you need to know on the outer segment. And again, we're going to look at both of these pieces in much more detail in a few moments. Down here, there's a sodium channel that stays open. So in the dark, you get what's called the dark current. Sodium channels are open. And they stay open with cyclic GMP. The second messenger. Remember, we've talked about how second messengers ultimately can result in opening of channels. So as long as there's cyclic GMP in the cell, the sodium channel stays open. What happens to the membrane voltage if sodium channels are open? Sodium's coming in.
Becomes more positive.
Exactly, it becomes more positive. So the membrane voltage kind of goes up. It's around minus 40 millivolts. So in the dark, so this is a little counterintuitive. In the dark, the rods and cones are positive, above threshold. Now, when they're above threshold, that means that they're actually gonna like these little, we talked about, remember in synapses, sodium, the calcium channels open and calcium comes in, and then you're releasing glutamate here. which is going down to the bipolar cells and all that. So in the dark, sodium channels are open because the cell is above this threshold voltage of -55. The cell is releasing neurotransmitter to the next layers of the retina. So again, this is a little counterintuitive. Normally, this would be easier to learn if it was just kind of asleep when it was in the dark and then it turned on in the light. But instead, it's going to be the opposite. So is this making sense? In the dark, we have a positive above resting voltage. And now let's look at what happens when light comes in. So we're going to focus in now on this stuff up here. So here's my membrane. I have my G protein, we're calling transducin. And this is my opsin. This is my retinal. All right, so retinal is kind of like a little antenna that absorbs light energy, and when it does, it actually changes its shape. So if you remember chemistry, if something is bent so two sides of the molecule are in the same direction, it's called cis. This would be cisretinal. When light energy comes in and it gets absorbed, it turns into transretinal. And transretinal detaches from the opsin and activates it. So this part right here, this is kind of similar to when we talked about a receptor binding a ligand and activating the receptor, and then would start the G protein mediated second messenger pathway. In this case, instead of binding a ligand, you absorb light. Retinal changes shape, detaches from the opsin and activates it. So now my opsin is activated. It's going to activate the opsin, the G protein. Transducin got its name because this was one of these pathways that was figured out. And they knew that it was helping transduce activates. They knew it was helping in transduction, so they called it transducin, but it's actually just a G protein. All right, so, and then what happens when you activate a G protein? Who knows, right? Tons of stuff can happen. You turn on a second messenger pathway. In this case, let's go back to our original picture here. So if light comes in, let me erase a little bit here so I've got more space. Okay, here we go. So now when light comes in, right, so retinal goes from cis to trans, activates the opsin, which turns on the G protein. Which is going to start a whole second messenger pathway. And ultimately, the result of that whole second messenger pathway turns on phosphodiesterase. which hopefully you remember. What does phosphodiesterase do?
Clears out the GMP, cyclic GMP.
Right, exactly. It's the stuff that breaks down that second messenger. It breaks down cyclic GMP in this case. So what's going to happen now? What happens if there's no more cyclic GMP around?
Would the sodium channels close?
The sodium channels are going to close. And then if the sodium channels close, what's going to happen?
Stop releasing glutamate.
Yeah, so now the membrane voltage is going to go back down to like the normal resting, and we'll stop releasing glutamate. So this, it seems a little backwards, but it's still a voltage change resulting from the stimulus, right? Normally the rod or cone is more positive in the dark. When light comes, it becomes more negative. So that's still, even though it's backwards from what you might have thought, it's still a voltage change that the retina recognizes as the presence of light. Right, and the circuitry of the retina, like I said, the retina is very kind of sophisticated neural circuitry. It has no problem just flipping the sign on that. It knows that, oh, going from more positive to more negative means, yes, light hit. So other questions, like, you are responsible for understanding this. Like if I asked you, like at the core, how does the eye do phototransduction? How do you convert light energy into an electrical signal? You need to understand this whole thing, that the light comes in and the light is absorbed by the retinal. As it goes to transretinal, it detaches from the opsin and activates the opsin, which turns on this G protein, which turns on the second messenger pathway, which turns on phosphodiesterase, which breaks down the cyclic GMP, which has been holding these sodium channels open. So as the sodium channels close, the voltage goes down, and there's our voltage change, our phototransduction. So make sure that makes sense. Are there any questions about it, like right now? So it's
I have a question.
Sure.
What does the glutamate do? I'm a little confused on that part.
Oh, it's just a neurotransmitter that talks to the next neurons in the retina. Like
Okay.
glutamate's just a neurotransmitter. So it's, I didn't mention, I should mention, so in the retina, we talked about there's like the rods and the cones. And then there are these bipolar cells. And there's like these ganglion cells, which are the ones that have the axons to be the optic nerve. And there's all these horizontal cells going back and forth in here. There's all these different cells. Except for the ganglion cells, none of these cells have action potentials. The ganglion cells are the only neuron in here. So all of the electrical activity is about voltages going up and down and becoming more or less positive, releasing more or less neurotransmitter. But it isn't until you get to the ganglion cells that you actually have a action potential which goes off to the brain. So these rods and cones here are releasing glutamate, which is binding receptors on the bipolar cells, which then is going to be changing the voltage of this, and that's more or less neurotransmitters over here. And ultimately, when the ganglion cell gets enough activation, it will ultimately send some action potential off to the brain. So, but yeah, the glutamate is just one of the parts of the communication between these different neural cells in the retina. So that's what its role is. So does that make sense? Okay. So any questions about phototransduction? And again, like I said, depending on what kind of opsin you have, it will maximally absorb light in different frequencies. That's how you get, like, rods and cones. They have the same basic thing going on. It's just what frequency of light they absorb is going to be a little different. And there's some other little differences as well. But for the most part, this is the basic story for both of them. Okie dokie. The last thing I want to mention about vision is just the idea of two eyes and binocular stereoscopic vision and all that. So, right, we talked all about kind of lateralization of the brain, like how the right side of your body is represented on the left side of your cortex. With your eyes, it's a little different. Each eye actually goes to both sides of the brain, but what's lateralized is your field of view. So, like, if this is you with your two eyes, and each eye is kind of looking at kind of the same view. Let's say, it's a view. There's like trees and whatever. Oh my God, the Golden Gate Bridge. So you're looking at the world out there. Your brain is in your head. The right and left hemispheres. What part of the cortex is processing visual information?
Your visual cortex
And where is that located?
on your occipital lobe.
Yes, that's on the occipital lobe. So what happens is this side of the world is kind of landing on one side of both eyes. The other side of the world, let me try to maybe, is landing on the other side of both eyes. So each eye, sees the whole world. And what's going to be lateralized in the brain is this part of the world is going to be, this half of your visual field is going to be processed on the opposite side of the brain. So this side of the retina on this eye and this side of the retina on the other eye both go to the same half of the brain. Similarly, the other half of the retina that responds to the other side of the world is going right. So this is where you get the optic chiasma. Because each optic nerve from each eye, half of it goes to one hemisphere, half goes to the opposite hemisphere. because each hemisphere is getting 2 eyes worth of information from each half of, from one half of the visual field. And from that, 2 slightly different views of the same scene, that's how you can kind of create this experience of 3-dimensional stereoscopic vision. And like I mentioned, there's a lot of animals that don't have their eyes like that. Like, again, if you are a mouse or a bunny rabbit or a sparrow or something, it makes more sense to just have your eyes on either side of your head. So then you have a much wider field of view. Right here, I get really good 3D vision, but it's easy to sneak up behind me. A mouse or a rabbit or a sparrow does not have as good 3D vision, but it's really hard to sneak up behind them because they can see basically 360 degrees. There's no part of their field of view that isn't covered by one or the other eye. So, what else do I want to say about vision? Any questions about vision?
It's a little off topic, but animals that are nocturnal or that can see in the dark, do they have more specialized rods or opsin that they use to see?
Yeah, so there's so many different kinds of eyes. So I would have to look more. I'm going to assume they would have much better like rod vision than we do. Well, there are a lot of animals that actually have whole different types of pigments that we don't have, so they see colors that we don't see. Like there's like the mantis shrimp and stuff that have, I think, like 12 different kinds of cones. There are some little animals that literally will incorporate molecules from the things they eat into building their photoreceptors, so they'll respond preferentially to the wavelengths that are going to help them identify their prey. So that like the story about eyes gets really complicated once you start going beyond just the human eye. And I have no doubt that even just cats and dogs have a different array of different kind of distribution of rods and cones or one or the other, but I am not as familiar with the details of that. So, but yeah, I have no doubt that it's pretty different from us because obviously a cat is way better at running around at night and not bumping into things. So they have much better low light vision than us. And then some animals that have eyes, they're not that useful for them for detail. Like a mouse. Mouse has eyes, and they can notice movement, but they don't really care. They're not going to read the paper. A mouse responds much more from its sense of hearing. It hears way, way better than you do. And it understands its environment much more from that. And all those animals with whiskers, those whiskers are a whole other sensory domain that we don't have a real ability to understand. There's whole parts of their cortex that integrate information from their whisker systems and kind of construct a model of their surroundings based on information coming from whiskers. Like, the big whiskers, there's little whiskers coming off of their, what you think of as weird looking eyebrows or whatever. But so other non-human animals have ways of understanding the world and sensing their environment that, we don't even have a way to have a basic understanding of, I think. So they have different kinds of eyes, but then they even have different other senses that help them orient themselves in ways that allow them to navigate much more gracefully than we do, in certain situations.